Good evening. Welcome to UT Brainstorms. It's nice to see everybody. Thank you for coming. We might as well start with parking because it is what it is. So first of all, last month there was a snafu, and we apologize for that. We had requested one thing, and they had signed us up for something else. So I know that many of you had to pay on the way out of one of the garage. So apologies for that. We do have vouchers. You should grab one on your way out, and you just scan it. I know that's what we told you last time, but we mean it this time. That was on us. Also, it turns out parking seems to have an infinite number of ways to throw us curveballs, and so tonight's there's a concert at Bass somewhere. So without telling us, you had to pay $15 to get in there. So if you parked at Bass or at San Jack, apologies, they didn't tell us that, and so very sorry. If you parked and didn't pay, take one of these just in case. Okay, they'll be available on the way out. You just scan it and then you can get out. Okay, so apologies about that. So my name is Mike Mock. I'm a professor in the Department of Neuroscience, and we put on these events as our way of outreach to share our interest and expertise in neuroscience. And I want to, how many of you are first-time brainstormers, by the way? It's fun so welcome. Okay, it's always fun. So some of you know this, but we have the word conversation in every Brainstorms title because that's really tonight's point. We'll have a presentation, but we'll have a long question and answer session. And part of the reason we do that is that that's the best way to share information, and partly because that's what's in it for us. So we learn a lot from your questions, and so the information exchange goes both ways. So thank you for that. Tonight's topic may not be relevant, but I just want to remind you, nobody here is a physician, and so we can't do any medically related questions. It's probably illegal. It wouldn't be good advice, and so tonight is not a medical night. All right. So the format is that our speaker will give us about a 35 or 40 minute introduction to the topic. He'll introduce his two panelists, and then they'll field, the three of them will field questions until we have to stop at 8:30. You have two ways to submit questions. One is through that website. Feel free, some of our experts have already done this. Feel free to submit questions even during the talk. We'll also have microphones running around. Raise your hand if you're more comfortable with that. We'll try hard to go back and forth between the two of them. We'll get as many questions as we can in as we go, all right? Any questions about the questions? All right. So, everybody hear that? If you were one of the people that were asked to pay on the way in, take a voucher, keep your receipt. If you email it to UT Parking Services, which you can find on the web, they will send you a reimbursement. Okay. They say they're sorry, but that didn't help tonight. So I mean, seriously, take one of the vouchers on the way out, all right? And if you've already paid, keep your receipt, all right? Okay, so the boring stuff aside then, it's my pleasure to introduce tonight's speaker, Dr. Harold Zakon. I think Harold's been, since just after World War I? Harold's been here a long time. He's had a long and distinguished career here at UT. He's an extremely well-respected neuroscientist, and he's also just a great guy. We all love him. He's retiring, and so we worked really hard to get him just this one last time, because he's real fun to listen to. So he'll talk, and then he'll introduce his panelists, and then you grill us with questions until we have to stop. Okay?
And we're going to talk about the poisoned brain, a conversation about how brains have evolved protection against poisons, or not. So let's start with this quote, which you probably all know from Macbeth. Eye of Newt. and toe of frog, wool of bat, and tongue of dog, adder's fork and blind worm's sting, lizard's leg and owlet's wing, etc., etc. So, this sounds like a bunch of scary and weird things in a strange witch's brew, and it actually would likely work because it's full of things that are poisonous. So eye of newt. Newts have a poison called tetrodotoxin that blocks nerve impulses. Frogs have all kinds of different toxins. In particular, I'll talk about one called epibetadine. Adder's fork refers to the puff adder and its forked tongue, which has poisons. And then tongue of dog stands for the hound's tongue, which is a plant that has toxins. So this would be a really potent mix if you actually took it. Now, many animals use toxins or venoms, and this is like a very small, like a short list of them. And you can see invertebrates, vertebrates, even animals you wouldn't have thought of. For example, platypuses have a venom. Shrews have a venom. A few birds have a kind of toxin. So it's pretty widespread. So let me first do a few definitional things. What's the difference between toxins and venoms? Now, when we say toxin, we could say, oh, pollution from this factory is a toxin that, is harmful. I'm trying to restrict myself here to biological toxins, so made by an organism. So a toxin is a biologically derived molecule. It's harmful or lethal. It may be sequestered inside an animal and not have a particular way of releasing it. So if that animal's eaten, the predator gets the toxin. Or it can be a component of a venom. A venom is usually composed of multiple toxins, and they usually come from a specialized gland with a specialized apparatus like fangs or stingers. So why am I interested, and why should you be interested in toxins? Well, first of all, let me say I wear two hats. I'm a neuroscientist, but I'm also an evolutionary biologist. And as an evolutionary biologist, to me, they're a fascinating example of evolution in action. We can look inward. We can say, well, how are toxins made? Where are they in the body? What do they do on a molecular level to the animals that have them? To the predators, maybe, who eat those animals, can they defend themselves? How do they work to hurt the predator? So we can look at physiology or molecular biology. We can also look outward and say, how does having a toxin affect an animal's ecology? And I'll give you examples of these. Now, as a neurobiologist, I can say toxins have given us in the past a huge amount of information and continue to give us information about critical workings of the molecules in the nervous system. And I'll give you some examples. They may lead to some therapeutics. Now, I have to tell you one more thing before we get going, and that is how neurons make impulses and transmit across a synapse. So you can see this nerve ending, and what's called an action potential or a nerve impulse is propagated down along the axon. Now what happens is there's a protein called a sodium channel. Those sit in the membrane and they open. And when they open, they let sodium ions that are positively charged inside the cell, because there's a lot more sodium outside than inside. So it just runs down its gradient. It diffuses into the cell, and that is what makes the impulse. So the impulse will continue to the end, and there, these little balls that you see contain neurotransmitter. And when the impulse reaches the end, those balls open up and release neurotransmitter that goes across the synapse and binds to something on the next cell, or if it could be a muscle. So a motor nerve can go to a muscle, or one neuron goes to another. They bind, and if this is an excitatory synapse, the receptor opens when the transmitter binds briefly, just a few milliseconds, and more sodium goes into the cell, making it positive. And that's what essentially an excitatory synapse does. Now, the problem is, if you keep letting sodium from the outside go inside, the difference runs down. You start putting the sodium inside, and then you can't get impulses anymore. So we need a way of re-establishing the difference of sodium between inside and outside. And there's something called a sodium pump. And that's like, you have a nice little fountain in your garden, and it runs all the time because the water that comes out gets pumped back in. If you shut off the pump, you're just going to have water, and not be able to have a fountain. So I've chosen particular toxins that act at these sites. Now I'm going to tell you 5 stories tonight. Monarchs and milkweed, newts and snakes, frogs and snakes, cobras and mongooses, and it is mongooses, by the way, not mongoose, scorpions and mice. And the first 3 are going to be with one kind of toxin, and the second 2 will be with a different kind. Also, you've noticed we're going to have 3 snakes, and it's a premature introduction here, but Harry Green, who's one of the world's experts, and not just at Cornell, one of the world's experts on snakes here, and so he's going to be a panelist. Also, Jeff Coleman, who's in the back, another panelist, is a graduate student here working on poison frogs. So we've got a good panel tonight. Okay, I need to tell you one more thing. One of the most common kinds of toxins are a class of molecule called alkaloids. And here you can see, for those of you who know anything about chemistry, they have a ring structure and a nitrogen. And plants make these to deter herbivores. But you have probably had alkaloids this morning. Here, a short list, a very short list of some of the alkaloids that we get from plants. I hope the one you had was caffeine, by the way. So let me tell you a little bit more. Animals that use alkaloids don't usually make them themselves. They have to get them from somewhere. And even if they store them, they usually diffuse around the body. So the whole animal's insides will be bathed in this toxin. So it might sound simple. All you need to do is put the alkaloid in. But in order to do it, 1, you need a food preference. If you're getting them from your diet, you need to first know what to eat. Then you need some system to take up the alkaloids, hold them in high concentration inside you, and finally, and this is where I come in, resistance. You have to find some way, in my case I'm most interested in molecular mechanisms, of being resistant to the toxin. So the first one I'm going to tell you about is something called glycosides. They're not actually alkaloids, but they're very close. And a lot of plants make them, like these plants. And the most commonly used one is called waban. I'm sure you didn't know how to pronounce that. So waban blocks the sodium pump. So it causes, when the sodium goes in, it can't come back out, and it ends up shutting off the neuron. Plus, these sodium pumps are in many different kinds of cells, and they're very important in the heart. So, monarchs and milkweed. This is an Austin thing, right? We love it when the monarchs come through, and a lot of you probably plant milkweed in your garden for them. Why? Why do you plant milkweed? Why do they need milkweed? Actually, the butterflies can feed on many different kinds of flowers. But here's the key. Females only lay their eggs on milkweed leaves. So right there is a behavior that they've evolved to only lay their eggs on milkweeds. Those eggs hatch, and the caterpillars eat those leaves. Milkweed contains glycosides. And monarchs store the glycosides in their body, and that is the caterpillars, and they even keep them in their bodies into adulthood. And these are a defense against predators because these glycosides work against all the other insects that would be trying to eat them. So notice here I say glycosides in the adults are mainly stored in the wings and the exoskeleton. Now also notice the adults and the caterpillars are very conspicuous. So this is a warning to predators, don't bite me. So this is a classic photograph from Lincoln Brower from decades ago. When I was a boy and reading Natural History magazine, I remember seeing this. Anyway, this is what happens. This blue jay eats a monarch and gets sick to its stomach. And you probably know if you've ever gotten food poisoning or something, you don't want to go near that again, that food. So they learn very quickly to avoid eating monarchs. So how do the monarchs protect themselves from the high level of glycosides in their bodies? Well, it takes only two changes, two amino acids in a critical place where the glycosides bind to the sodium pump, two small changes, and it no longer binds. So that is how the butterflies, the monarchs, protect themselves. But that raises a question. If a toxin-carrying animal can evolve resistance, can a predator evolve resistance to the same toxin and overcome the prey? And in evolutionary biology, and this is fitting this week and who knows how much longer, we talk about it as an arms race because you have some weapon and you come up with a countermeasure like an interceptor for a missile. So in this evolutionary arms race, occasionally some animal will be able to crack the code, basically. And so here, most birds will not eat monarchs because they're not resistant, but a few species do. The sodium pump of these birds has, just like the monarchs, independently evolved some of the same amino acid changes in the protein that keep the glycosides from binding. In addition, these birds also have behavioral adaptations. This, even though I work on molecules, we must not neglect behavior. And in some cases, like in this case, the birds learn. Remember I told you the toxins in the wings and the exoskeleton, they learn to pull off the wings and slit the abdomen open and eat their guts, basically. So they have learned how to minimize whatever toxin they would get anyway, as well as being resistant. So I just want to say, having a predator like this be able to attack them can have major ecological consequences, and I will tell you about them. It's estimated that 70 to 40% of monarch deaths in the overwintering populations in Mexico, up to 40%, are from grosbeaks and a few other birds that have figured out, evolved and have behavioral adaptations to get them. Now, these numbers come from Lincoln Brower, who is a pioneer in monarchs, and Bill Calvert, who lived in Austin and led many expeditions down to the butterfly overwintering grounds. I know at least one person who's been down to that site with Bill Calvert. Austin Institution. Okay, so here's my summary. Apologies to Eric Carl. The milkweed makes the toxin. Most insects can't eat it, but the monarch, this blue arrow means it can take it in, can. It metamorphoses. Most birds won't eat the monarchs, but the rose-breasted grosbeaks eat the monarchs. Okay, humans have known about this for millennia. In Africa, where some of these plants grow, they use it on their spears to kill hippopotamuses, I mean, big animals. It's a very effective toxin. Folk remedies in Europe using related plants like foxglove have given rise to digitalis that hopefully none of you have ever needed, but is a heart medicine. And this last thing really blows me away. It's controversial, but there's a claim out there that we may be able to produce Waban ourselves in very low quantities, and it can aid in regulating blood pressure. So that's what we know in terms of humans. Now, next story, newts and snakes. Here is a toxin that blocks sodium channels. And it's called tetrodotoxin. The rough-skinned newt makes it. TTXs throughout their bodies. When they're attacked, they have it in skin glands, and it's secreted, so a predator gets a bunch of it in its mouth. It only takes, again, this is what's amazing to me as a molecular neuroscientist, it only takes a few changes, subtle changes in the sodium channel to prevent the tetrodotoxin from binding. So the newts, of course, had to evolve channels that don't poison themselves, don't get poisoned. Now, I just have a very brief video I want to show you, and I don't want to go, I have a few other things, so I don't want to go too long over, but I think you'll like this video. We'll see if it works. If not, I'll use my microphone. I don't think it's working. Oh, where'd it go? No voice. Okay. The soundtrack says there's a bullfrog going after that newt. It eats the newt. Is it going to be a yummy dish for the bullfrog? Let's see. The bullfrog collapses and dies. But what's that? The newt climbs out unscathed and heads for home. I guess I'll have a career as a narrator. There was this voice track and somehow it disappeared. Anyway, so garter snakes can eat newts. Garter snakes, and they're all across the country. We have them here, but only the populations of garter snakes that overlap with these newts that live on the west coast, basically from British Columbia all the way down into Canada. And ones that are hundreds of miles away didn't evolve, and don't overlap with the newts, didn't evolve the ability to eat them. So how do the newts do it? Well, the newts are tetrodotoxin resistant. The garter snakes do the same thing. They have channels that are not blocked by tetrodotoxin. Most other snakes can't, but these garter snakes can eat them. But there's good news and bad news, because the molecular changes that protect the snake from TTX come with a cost, because the sodium channel, in order to prevent the tetrodotoxin from binding, has something that happens to it that makes it less effective at its job as a sodium channel. And if it's in the muscle, it makes the muscle not work as well. So a snake that doesn't have TTX, who got, normally is faster. One that had the mutation to protect it is slower. And if that mutation happened in a snake that didn't have TTX, it would be slower. It couldn't catch prey, might have trouble escaping. So it's worth a cost. There's a balance. If it can protect itself, if natural selection allows it to be able to have this mutation, so it doesn't have TTX bind, but it comes with a cost, and the benefit outweighs the cost in their evolution. But there's more. You probably know fugu, a pufferfish, where the Japanese first discovered and figured out the chemistry of tetrodotoxin. It's a delicacy in Japan. But TTX is found in many unrelated animals. Blue-ringed octopus, very poisonous. It squirts out TTX on its prey to paralyze it. All these different animals. How? How do all these animals have TTX? Well, TTX is actually made by bacteria. And all of these animals independently set up a symbiotic relationship with the bacteria. So the bacteria live inside them, provide the TTX that protects them. Those animals all had to evolve resistance in their sodium channels. And it's a nice symbiosis. So the full story: bacteria, the newts, and the snakes. Now, the medical or the human angle is tetrodotoxin is very useful for humans in some circumstances, and it is used therapeutically, especially in patients who are dying of cancer and are in pain. It will block the pain. We can talk about it later because it's a tricky thing to use. Okay, poison dart frogs and royal ground snakes. South American poison dart frogs accumulate alkaloids, but they get it from their diet of ants and mites. So once again, the animals have to want to eat ants and mites, and they have actually specializations to scarf them down quickly. They also have bright colors to warn predators. Here, the toxin that they have, I'll talk about one called epibatidine, blocks the receptor in the postsynaptic neuron. So epibatidine acts just like the normal neurotransmitter called acetylcholine, but it's 1,000 times more potent. It was discovered by John Daly down in South America, who was studying these frogs. Now, this study was done here at UT. Dave Canatella, who's the chairman of integrative biology, and I talked about this for years. Becca Tarvin, who is our joint graduate student. Lauren O'Connell, who is here for her PhD, went to Harvard, and then she's now at Stanford, contributed to this. And Cecilia Borghese, who I thought I saw out in the audience somewhere, contributed, there she is, with this as well. So this all-star cast worked on this problem. So it turns out a single amino acid substitution, that's all it takes, makes their receptors resistant to epibatidine. But it causes a decrease in the number of receptors because it, we think, disturbs their ability to form and get to the cell membrane. So this is just another example of a trade-off. It's worth it for them to have this. So we have a bunch of experiments underway to try and figure out how they are able to successfully live with low numbers of these receptors. How do they compensate? We don't know the answer yet. So Becca Tarvin, went off to her job at Berkeley and published a paper from her lab just last year. They found that while most snakes will not or cannot eat them, the royal ground snake does. It still can be killed by them, but it has evolved a behavior where it grabs the frog, wipes it on the ground to wipe off the toxin, and then swallows them. Now, if they give them non-toxic species of frogs, they eat them. They don't wipe all the frogs, so they maybe taste the toxin or something, but they have a behavior to get rid of the toxin. So once again, behavior saves the day. Now, again, one quick little piece. I don't know if there's going to be a voice, but this is about a related species, not the ones that we study. These frogs are amazing. They lay their eggs in bromeliads, and they come back every so often, and the females drop unfertilized eggs for the tadpoles to eat. But there's more than just that. I'm going to see if I pull this out what happens.
She gorges on tiny, poisonous ants. But this ingenious frog can assimilate ant toxins into a much more potent compound, making her skin fatal to the touch. Now she needs to pass her poison power to her young. In each poo, she lays a single unfertilized egg laced with neurotoxins. And as the tadpoles eat, poison releases from the egg and floods the nursery pool.
And this is why I don't use technology. Okay. Okay, so here's the big picture for the frogs. They eat ants and mites to get the toxins, and then most snakes and birds are susceptible and won't eat them, but this one species evolved a behavior that allows them to. Now, this question mark you can bring up with Jeff Coleman later. Do the ants and mites make the toxin, or where do they get it? So it could be a whole food chain. Okay, humans, of course, have known about these toxins for ages, for millennia, and that's why they're called poison dart frogs, is the natives use them in their darts for hunting. Now, epibetadine, the toxin we work with, was developed as a painkiller, but it had too many fatal side effects. I like to say that the difference between killing the pain and killing the patient was too small. So they just stopped using it. But sadly, it's been in the news. I don't know if you saw this. You probably know about Alexei Navalny, who died a few years ago, and just a week or so ago, it was discovered that it was epibetadine, this frog toxin, that killed him. But the good news is it's very important. You can see epibetadine looks a lot like nicotine, and epibetadine has been a very important reagent to study things like nicotine addiction or epilepsy, because these receptors are involved in epilepsy. Now, I'm going to quickly tell you two more stories with a different kind of toxin. These are peptides, which are strings of amino acids. Unlike the alkaloids that get taken up, the animals make these. They have genes to make them. And then they have specialized glands that express the genes that make the toxin. So the toxin isn't everywhere. It's only in these glands. And they have specialized fangs or stingers that have evolved to deliver the toxin. And the venom usually has multiple components. So the first one I'll tell you about also binds to the receptor. The one from the frogs opens it up and makes it a super active. And it's too much neurotransmitter. The one from snakes binds to it and prevents the neurotransmitter from acting on it. And in particular, the cobra toxins that I want to talk about. Cobras, even though you would think they don't need to be, are resistant to their own venom. Why? Well, I will explain. First, what prey fits best inside a snake? And I heard a number of people say another snake. Good for you. A+. Another snake. Cobras, especially king cobras, specialize on eating other snakes. And they love other cobras. So cobras had to evolve resistance to their venom. In some species of snakes, when the males fight for dominance, they don't bite each other. Their behavior has evolved so they don't use it against each other. And they may not have, rattlesnakes, for example, much immunity to their venom. But cobras had to have this because they will eat each other. Of course, a mongoose can kill a cobra. And so the bottom line with the mongoose is cobras hunt other snakes. Most of them are susceptible. Cobras are not susceptible to their own venom. And the mongoose cracks that by being able to attack the cobra. Now, snake venoms have been very, very useful in mainly studying the neuromuscular junction, where the nerve synapses on the muscle. That's where many of the snake toxins are directed, to paralyze their prey. And let me just show you one thing from a lab. This shows the particular snake toxin labeled with a fluorescent molecule that binds at the transmitter receptors here on the surface of the muscle. There's a protein called dystrophin because it was the protein that was eventually found to be mutant in somebody with muscular dystrophy. And you can see if you overlay these two views, and the dystrophin is also labeled with a fluorescent molecule, but it fluoresces at red instead of green. You put these two images together and you see they overlap perfectly because the dystrophin, part of its job is holding the receptor in place right below where the nerve is putting in neurotransmitter. And in this knockout mouse, that is a mouse where somebody destroyed the dystrophin gene to mimic what happens in a patient with muscular dystrophy, you can see the nice order of the synaptic site falls apart. And that's what happens. Without the dystrophin, the neuromuscular junction begins falling apart, and the ability to command your muscles to move is gone. I'm going to end with one last example, and this is, again, something we've done in our lab. So scorpions subdue their prey with lethal stings and strong pincers. Here's a hornet. And, clearly you don't want to get stung with this. Another thing that's interesting about their venom is there are many different peptides. And because the insects have sodium channels too, but they're a little bit different from ours. So they have evolved a range of peptides. Some work only on insect channels and not on us. Some work on ours, that is mammalian channels, and not on insects. But they have a whole gamish in their venom gland, and whatever bothers them, if it's prey or a predator coming down for them, they get stung. And in that mix, there'll be toxins that target their channels. Now here's Ashley Rowe. She was a postdoc in my lab for a while, pioneered this system, and she's now at the University of Oklahoma. When she was a graduate student elsewhere, she discovered that grasshopper mice eat scorpions. And now this map shows in Texas, we have a species of scorpion, which, how many of you have been stung by scorpions? Yeah. Was it pleasant? Yeah. Well, the one in Texas is painful, but it's not going to kill you. The one in Arizona, the Arizona bark scorpion, is potentially lethal to human beings. It has the strongest venom. And yet, as I'll show you, the grasshopper mice eat them down like candy. Now, why would a grasshopper mouse eat a scorpion? Well, they live in the desert. There's not that much food there. So anything you can do as a source of food is great. And if you can crack the code and have at those scorpions, you have a food supply nobody else has. So they specialize on these scorpions. Now, I'm going to show you another video. This doesn't have sound, so we're okay. Just watch this face-off that Ashley made between a scorpion and a grasshopper mouse. Just stung 3 times in the face. And he goes right back, kills it, and eats it. Now, if you were stung by a scorpion, the bark scorpion, 3 times in the face, you would be howling, waving your arms about, and saying many unmentionable words, no doubt. And here's the cool thing. So scorpion toxins open the sodium channel. And there's a special kind of sodium channel in our pain receptors. And that toxin goes right to those and opens it up. And that's why we feel pain. But with just a few amino acid substitutions in the sodium channel of the grasshopper mouse, what it does is instead of the toxin binding and opening the channel, the toxin binds and keeps the channel closed. So the channel has evolved a way of making a painful toxin into a painkiller. When we sent this paper off to the journal for publication, we called it Evolutionary Jiu-Jitsu, using your opponent's strength against you. Unfortunately, the editor said, that's a cutesy title, get rid of it. But oh well. So here's the grasshopper mouse's superpower, which is the channel has evolved to take something that opens it and make it into something that closes it. So it can't feel pain. Okay. Now, and as we speak, Ashley in her lab and her collaborators are starting to work on using, studying the interaction between the toxin and the sodium channel of pain receptors to see about engineering it as a therapeutic for blocking pain. So I just want to sum up. Toxins and venoms, I think, are an exciting topic in studying evolution. We can look inward at the adaptations in physiology and molecular biology that the animals have. We can look outward for its role in influencing an animal's ecology, and they're very important for studying the brain and as leads for therapeutics. So thank you for coming. Yeah, so I introduced them briefly. Harry Green, world expert on snakes, reptiles in general, but snakes are his thing. He's published some great papers and written books on his amazing expeditions and encounters. Jeff is a little earlier in his career, no books yet, but he will someday be writing books on his incredible career, and he focuses on frogs.
Okay. Is this working? Yep. Okay. So why don't we start with the, I'm going to start with a question here. Raise your hand if you'd like to ask a question without the computer. We've got two students in the back who will try to find and run down to you. But I'm going to read the first question here, which was given while you were speaking, in fact. The question is do gut bacteria and parasites have venoms, or are they toxic?
Yeah, I suppose I should say something about it. Well, my general answer is I don't really know. But there are plenty of bacteria that make toxins. I will say, if we looked into the bacterial world, it's every bit as vicious as everything I've been talking. Bacteria eat other bacteria, they have toxins for each other. Yeah, a lot of toxins that bacteria have. Whether they're made by ones that colonize our guts, I don't really know.
Yes, please.
I'm a little nervous. But a few years ago, I got bit by a scorpion here in Texas, and I thought I was about to die. And like literally, anytime I would think about the scorpion biting me, my foot would hurt because it was. So my doctor said that the darker scorpions are more lethal than she was like, well, if you were in Africa, it would be an issue. So like, are you saying that like we do have scorpions here that could be lethal?
Well, I guess I'll take that. Unless. Okay. Okay. Well, the rule of thumb, I'm not an expert. Ashley in my lab was an expert, but the rule of thumb is the bigger the scorpion is, the less toxic.
Okay.
Because when they're hunting prey, the bigger ones rely more on crushing them with their, pincers,
Yeah.
right, thank you. It's the smaller ones that really need the protection from the toxin. So it's size more than anything
Okay.
that correlates.
Gotcha. Thank you.
Thank you. Yes, of course. You did it.
How about some questions on frogs or snakes?
What's your favorite snake? Yes.
Is this working? I've been asked that question so many times, and one of my best friends here on campus teases me about having so many favorites of any category you name. So I have my favorite longhorn, I have my favorite frog and toad, I have the favorite individual snake I ever looked at, which was a female black-tailed rattlesnake I studied for 12 years in the field in Arizona. But I really like, here in Texas, I think broad-banded copperheads are some of the most beautiful snakes in the world. There's a start. And actually, I'll just say one more thing because it ties into what was talked about tonight. There's a snake that's found only in the Texas Hill Country. It's a subspecies of the black-necked garter snake. And unlike the more wide-ranging black-necked garter snake that's in West Texas all the way to Arizona and down to Mexico, our hill country black-necked garter snakes are gaudy. They're just spectacularly colored with bright yellow, green, and orange, and so forth. It's so unusual that it begs the question, what's special about them? And one thing Harold didn't mention is that it turns out that garter snakes out west that eat newts tend to be more brightly colored. And so once I started thinking about that, I started looking into the diet of our hill country black-necked garter snakes. And it turns out they eat a lot more toads, which have toxic skins. And so nothing proved yet, but my bet is down the road, some graduate student in your lab or somebody else's is going to find out that our spectacularly beautiful black-necked garter snakes here in the hill country are very special.
Jeff, your next project.
We've got another from the app. Are there any toxins from a snake or frog that can stay in our systems and cause mischief later?
Do you?
Yeah, I'm not sure. I know that. I mean, yeah, I feel like this whole situation with Navalny, I feel like if it's at a lower dose, it can probably stay for a while if you keep dosing it and your body isn't clearing it that quickly. and over time that can be potentially lethal. But I'm not sure about something where our glands just store it for a long time.
Another snake question. Sea snakes have been, at least the ones I've known about, like in Australia, you don't swim on the beach because there are sea snakes that are quite poisonous. Can you tell me if that's the same kind of system as some of the land snakes, or do they have a different type of toxin?
So Harold might want to chime in. What I can tell you is that sea snakes are closely related to cobras. They're in the same family. And so in general, their venoms are very heavy in terms of neurotoxins. That's about as far as I can go with it.
Yeah. Don't know.
All right, well, here's a question from our favorite Brainstorms audience member from Westlake High School. So.
This is really a shot in the dark because I know nothing about venom at all, but how do, like, antivenoms that you would administer to someone who has been, like, bitten by a snake or something venomous, how does that affect, like, your neuron and the synapse? Like, what does that do to reverse the process?
Okay.
That's a great question for Harold.
Okay.
I was going to say.
Antivenoms, so they basically are antibodies that bind, and antibodies bind to things and then, will pull them away from their target. So the general idea is they will bind to the venom that's circulating after a snake bite and try and keep them from getting to their targets. One encouraging piece of news, well, two things that I read about recently. So each venom, you need to make antibodies specifically for that venom. So it's very specific. There are people like this in the world, and Harry may be close to people like this. I don't know. People who study snakes are crazy. They will handle the most dangerous animals. But there was a guy recently who built up immunity by letting himself be bitten by different species of snakes. And so because he had so many, and he planned it to go across the family tree of poisonous snakes, so that his blood, his plasma, was full of all these different antibodies that could now function more generally for almost any species of snake. Yeah. Buy some stock in his company. Well, I think that is the idea, is they would, get some of his blood, get the antibodies, and then be able to give it to a patient, someone who is bitten.
So here's a question about evolution. So what's the time scale? How long does it take species to develop these resistance to the toxins and this dance back and forth where they're adapting? Were you asking me? So, yeah, okay. It depends, of course. Now, the thing is, what I find remarkable is it only takes, of the examples I've given, it only takes 1 or 2 or 3, a very small number of amino acid changes. And if you think about it, all the time, this is happening. I mean, I could take probably any protein of everyone in this audience and we can compare them, and there'll be a few amino acid differences because that's just what happens over time. Genes change, very slowly, and some of the changes have no effect. Some of the changes cause diseases,
and you might die. So that molecular change is not passed on. Any molecular change that's good, this is basically natural selection in action, will be selected for. So given that, it's very likely that there's this sort of variation in populations, and there's a normal rate of change in genes and the proteins they encode, it might not take very long. So on one hand, think about all these different animals that have TTX. They all have channels that are resistant, but not every animal does. So, it's hard to say how many years or how many generations it would take, but it doesn't take much. So it can happen relatively quickly. But I will just say this. The genes that snakes, for example, have for the toxin peptides, those evolved really fast. So they're constantly changing a little bit the toxins they have. And if they can come up with something that their prey can't match right away, then the snakes are on the winning side. And then it takes a little while. The prey may or may not evolve changes to be able to withstand that. So it's a constant battle, really, between the predator and the prey.
It's my understanding that decades ago, the blue indigo was imported from India to eat the rattlesnakes, but they're saying that it's indigenous down in South Texas. Did it become indigenous because of the importation?
Actually, I've never heard that before, and it's not true. Indigo snakes, sometimes called blue indigos, are found in the U.S. and Florida and Texas, well, in the southeastern U.S. and in Texas. And they're a very wide-ranging snake all the way from Argentina to up here. And they're one of the biggest non-venomous snakes in the world. And one of their two favorite food items are other snakes, and they readily eat rattlesnakes. So it's not uncommon on social media to see scenes of indigo snakes in South Texas eating rattlesnakes. But they're totally native. I mean, we have a guy in our department who's even older than I am who grew up in South Texas, and he remembers that in South Texas schools they were encouraged to let indigo snakes live around the playgrounds because they knew they kept down rattlesnakes.
All right, we've had several questions that all related to how did you guys get interested in these topics? So it's a question that all three of you could deal with. So,
Well,
what got you interested in the first place?
Yeah.
Let's hear from you.
Yeah, so I'm not sure my longstanding interest was necessarily in how animals acquire toxins specifically, but I think what initially got me interested in just broadly in evolutionary biology was just my childhood love for the outdoors and just being in nature, especially with my dad. He and I would go out to I'm from the Chicago suburbs, and we'd go out to the forest preserves a lot around Chicago. I think just the excitement of seeing that kind of different animals lived in different places, like you'd go visit Florida and you'd see all the anoles and stuff, and then go back home to Chicago, and just the most exciting thing would be like a garter snake or a corn snake. I mean, it was interesting in that sense. So I think that's what got me started thinking about wanting to do a career related to natural history. And then in high school, I got really into like chemistry and math and a lot of more technical aspects. And then I think in college, I did my undergrad at Berkeley, where there are a ton of herp people and snake people. And when you're telling that story about snake folks being crazy and grabbing it, I'm thinking of one professor in particular who had no problem grabbing rattlesnakes on our field trip and showing off their fangs. You might know who that is, but yeah, I think just seeing that you could, there was a whole world of kind of chemistry and mathematics and all these things that felt very technical that you could kind of apply to this world of natural history and being outside, because I started taking classes and doing lab research and finding that you could kind of marry those seemingly pretty disparate dimensions. So I think that's kind of like a long story of how that unfolded for me. So yeah.
what words of wisdom or caution do you have for kids who might pick up snakes and frogs at random?
So I'll just say real quick, there's now a professor at Berkeley who's Becca Tarvin who was mentioned in this talk. She did a lot of this research, and she remarks on this faculty member that I just alluded to. None of us thinks that's safe. And I mean, it's great that he does it. He knows what he's doing, but don't do it at home. You know, I wouldn't. Yeah, I guess unless you're trained. Harry knows this, but that was just my own.
So, but you were asking about the general population, right? What would we tell anybody? And in terms of snakes, I would say you should never pick up a live snake unless you're absolutely sure that it won't hurt you. The second thing I'll say is that even the venomous snakes, once you've seen that snake, it's essentially not dangerous to you anymore unless you pick it up. I mean, there aren't, I think I can really say this, there aren't any venomous snakes in the world that I think are truly aggressive that want to attack people. It's all defensive. And so your job is to not make that coiled up rattlesnake that you just stepped on or might have stepped on think that you were going to hurt it. So I think we can do a lot to minimize danger, and one of them would be to just never pick up something unless you feel comfortable, unless you know that it's okay.
What are the 4?
Pardon me?
What are the 4 snakes that are aggressive? Where are they?
Oh, I didn't mean to say 4. I don't think there are any. Even famously, supposedly aggressive species like black mambas, people I know who work with black mambas a lot in southern Africa say that give a mamba a chance, it'll always get out of your way.
We've heard a lot about autism and vaccines, and I've seen a lot where snake venom was used in a lot of those vaccines. Any thoughts?
I've never heard of snake venom being used in vaccines. I'm not an expert on vaccines, so I can't
That's
say
just where
for
I've read it.
sure, but I just don't know.
Just thought I would
You
ask. Might be something somebody could study sometime. I mean, because I have read it.
okay, well,
So there were a couple of questions about, are there toxins that have psychedelic effects?
oh, definitely. Licking toads. Oh,
No, I wasn't. I wasn't implying that I've experienced it before, but I'm very familiar. Some of the tree frogs, the Phylomedusian, like if people have heard of combo, that tradition of like licking the frogs, that's where it comes from. It comes from this one group of tree frogs, and those are not diet acquired toxins. Those are toxins the frogs synthesize themselves. They're simpler peptide toxins, I believe. Do you know about that? Do you know about combo? Oh, okay. Yeah, so I've read articles about it, but no, I've never. I know it gives you quite a trip, but yeah.
well, and you know,
Allegedly.
just to say, we are so weird. You saw that list that I made of alkaloids? Those are to get rid of things. Like caffeine is supposed to be insects start eating it and like, ooh. But humans, we love this stuff. It's so weird. Like the second thing on the list of alkaloids was capsaicin. Anyone know what capsaicin is? Yeah. Where does it come from? Anyone know? Capsicums, right. Chili peppers. Mammals, small mammals, or mammalian like rats, etc., will try and eat those, and they go, "No, thanks. This is burning my mouth." But look at us. We humans, we love to eat it. So same with the psychedelics. They're meant to cause an animal to, if they grab the toad, to go, "Oh, what's happening to me?" And then never go after it again. But people are so weird.
We live in the hill country. There are a lot of snakes. What are some simple rules of what you should do, or whether you make noise or whether you put on certain scents to make the snakes head the other direction?
I don't think that's one of the things that's going to work, making a lot of noise. The way to look at this, I think, is to talk about and think about how people do get bit. And the very first thing is to just not simply pick up any snake unless you know it's not venomous. The next thing is if you're going to be out in high grass and brush, wear boots or wear snake chaps. Right there, we could probably prevent most venomous snake bites. The next thing would be, and I ranch longhorns in the hill country, so I'm out there all the time and I'm worried about things other than snakes. So I know that it's a little bit challenging cognitively to deal with various different issues, but you want to always be watching where your hands go. You don't want to be sticking your hands under a piece of plywood or sticking your foot under the edge of a shed where a western diamondback could be sitting out the day. And the last thing is if you're out at night, you want to have a flashlight. And especially, don't go out barefoot at night without a flashlight. The most recent snake bite I know of in Mason County, where I live, was somebody who was walking around in flip-flops at night without a flashlight. They stepped on a copperhead and blamed the copperhead. So I've been around venomous snakes my whole life, and I've only been bitten once, and that was when I was 17 by a snake I picked up. Since then, I've worked with venomous snakes all over the world. I'm always very careful, always very respectful. I have rattlesnakes in my yard. I don't even worry about them.
I've heard that the rough-skinned newt in California is probably the most toxic animal in North America. I was wondering if that's true, what would you put as going down the list of toxic animals from that to other things?
Yeah, that was my undergrad work. Part of that was on those Tarika newts. I think because tetrodotoxin, I think it's one of the alkaloid compounds. It's one of the most potent. I think. After we didn't touch on this tonight, but there's this toxin, batrachotoxin, that is sequestered by a group of poison frogs that live in Colombia and Panama, and that's like the most potent alkaloid known to science. And those frogs are incredibly toxic. I mean, just licking them can be lethal. That's not exaggeration. But I would say that's probably true that the Tarika newts are the most toxic. Tetrodotoxin is really, really potent. But the Tarika newts also the Notophthalmus, the sister genus, has it too, which live out east. So the Tarika aren't the only ones in the United States that have them. But the compound itself is up there. Yeah, and I would touch them with my bare hands. Probably not brilliant, but yeah, other scientist mistakes there.
Can we put not licking newts as part of your guidelines for?
Yeah.
Yeah. That I don't know that I would have needed to be told that, but
yeah.
So it seems like environment plays a major role in the development of these genetic resistance. So my question was, does genetic resistance to poison adaptation have like an age restriction? Like, is it better for these things to develop the resistance in early development, or does it have more of a tolerance built in the mature stages of life?
Well, I'm sort of winging it here, but I mean, in general, if you're talking about using venom, for example, or having a toxin, so for most vertebrates, the bigger you are, the more you can have. So smaller animals are usually less venomous or less toxic than bigger animals. Also, in the case of the monarchs, they start sequestering, the toxin when they're caterpillars, and they keep it with them into adulthood. So it may be that the adults have more toxin in them. I'm not really sure. Certainly, in the case of frogs, the ones we talked about, they have to eat ants and mites that have the toxins, and no one's actually asked Jeff yet where they get them from, but the more they eat, the more toxins they'll have. So, yeah. It's not something I've studied, but that's my off-the-cuff answer.
So where do the ants get them?
I was going to follow up on that point, but yeah. So where did the ants get the toxins that confer the toxins to the poison frogs? I don't think we don't really know the answer to that question. You know, you mentioned that I'd be able to give folks a satisfying answer about that, but I'm not sure. I think in some cases it's suspected that the microbes that live on these small ants and mites might, for some species, be synthesizing it for them and then living on the surface of the arthropods. And then I think in other cases, I mean, we're just starting to sequence the whole, the entire DNA sequence of these animals called the genome, especially of a lot of these small invertebrates where it's really hard, and there are a lot of regions of the DNA sequence that are really tough to sequence. But researchers are starting to find pathways that might potentially be involved in the production of some of these toxins. So some arthropods might be producing it, maybe more mites than ants, at least in the poison frog story, it seems like. But it's really speculation at this point. Yeah. Hope that's helpful.
So there was a question online that went something like this. So antitoxins work by triggering antibodies that attack the toxins. Are there any natural defenses that work in somewhat the same way?
Oh, yeah. I didn't talk about that because, yeah, there's too much to say. But many species of snakes, for example, have evolved blood. So we have all kinds of proteins floating around in our blood that do various things. And one group in general are called proteases. Those proteins cut apart other proteins. And so a lot of species, for example, I think they're wood rats out in California, can be bitten by rattlesnakes, and nothing happens to them. And their defense is very different. You know, as a neurobiologist, I'm interested in channels and things like that, but they have proteins that have evolved to have proteolytic activity. So those proteins will cut the venom into pieces. And so that's another kind of defense. There are many other sorts of defenses than the ones I've talked about. So
Yeah, I mostly had a comment when you were talking about using some of these blocks for painkillers. And last January, Vertex got FDA to approve the first sodium ion block of channel block, which I just had for knee replacement and worked amazingly, called Journevax.
Oh.
And it's a non-opioid. You don't get high, you don't throw up, and you can't get addicted, and it blocks moderate to severe pain really well. So this was really interesting for me because I wanted to know how, of all my friends who said this was a surgery from hell, I managed to do it pain-free. So I really appreciate your talk today.
Yeah. Well, this is very exciting for me to hear this. A former postdoc in my lab took a job with Vertex, and her project was working on this. And so I was communicating with her. Of course, she couldn't tell me anything because it's top secret. But when they were developing this drug and then started it in clinical use, I've followed it, you know, over the years. And when it came out just a year or two ago, I was waiting to hear of people who used it. I tried to convince my sister to get it.
I was the first one that
for back surgery.
My knee surgeon I told him I wouldn't do the surgery without it. And I was just at PT this week, and two of his patients next to me said he'd put them on it. So it's just starting, but you have to push for it because it's called Journevax, Journavx, and the pharmacists don't know about it. I used it with a periodontal surgery this summer, which made me think I could do knee replacement because that was a bad surgery. I've had friends I talked into it with wrist and hip, but you have to push for it because they haven't used it, and they don't want to be the first ones out of the box, as my surgeon said. But now he's telling other people, because the alternative to opioids in a long surgery recovery like I have is huge.
Yeah. And that's the goal, non-opioid painkillers.
So buy stock.
On another note about the commercialization of this research, one of the elements in the presentation, which I didn't quite catch some of the text about, was the TTX used for neuropathic pain and as a possible new avenue for using these products. Can you tell us anything about how that works? Because neuropathic pain is such a reason for people to commit suicide.
Yeah.
And
Yeah. Well, so that actually is a great follow-up to the last comments. So finding a non-opioid painkiller has been, I would say, the holy grail of pharmaceutical companies for 30, 40 years. One of the problems is with using something like tetrodotoxin or things that block sodium channels. We have 10 different genes for 10 different kinds of sodium channels. There's one kind in the muscle, a different kind in the heart, different kind, in different places, like in different ones in the brain. And they're so similar that any drug like tetrodotoxin that blocks one will usually block the other ones. So it's very, very difficult to use tetrodotoxin because you're blocking everything. And so the particular sodium channel in the pain sensing neuron is very unusual as far as sodium channels go. And the difficulty was getting tetrodotoxin-like substances to work on it because they don't bind it. So it was trying to find anything new that could work on this channel. And the people at Vertex gradually, and like I'm saying, this is 30, 40 years of failed experiments, but it finally ended up people at Vertex came up with a new compound, and that's what we were just talking about, that works on acute pain. Now, the reason I say acute pain is they took that drug and tried it again on chronic pain like you would get with neuropathy, and it didn't work. So they're back to the drawing board to try and tweak it. And these things take years to, tweak something and try it in animals. And if it works, you try it in people. You've got to go through 3 different clinical trials. First make sure it doesn't kill you, et cetera, et cetera, et cetera. So it takes years for these drugs to be developed.
So one from the app. So are there instances where animals are using toxins for advantageous reasons other than defense?
You know of any?
Yes, actually, that's probably the primary. Is this working? Yep. That's probably the primary purpose of venom in snakes is to subdue prey, not for defense.
Oh.
So, and the kind of evidence suggesting that's indeed true is that we took species of rattlesnakes and species of coach-whips and whip snakes that lived in the same place and ate the same prey, and we just asked the question, do the venomous species get heavier prey items per event than the non-venomous species? And the evidence was very strong. On average, when sidewinders eat western whiptail lizards, they get a much bigger prey item than when coach whips eat western whiptail lizards. So, yeah.
Yeah. So I wasn't sure if the question was beyond defense and predation. I don't know. Was the question, do they get some other benefit? Was that the question?
That's how I took it. I'm sorry if I took it
No.
wrong.
I'm glad you did. I wasn't sure if they were asking, is there anything else beside these kinds of interactions between predator and prey? Yeah.
Okay, who'd like to ask the last question? Okay, here we go. So last question of the night. Thank you.
A snake question. Between, like, some snakes have, I understand, neurotoxins and some are more hemolytic in their effects. Is one more advantageous in the type of prey they eat than the other?
That's a great question. What I can tell you is that the species, and this is mostly vipers that have what sometimes are called hemotoxic or tissue-destructive venoms, they tend to eat really heavy, bulky prey that could actually rot inside their stomachs before they digested it. So we're talking about eating something that weighs 150% of the mass of the rattlesnake. So just imagine me taking on a 225-pound cheeseburger, somehow getting it into my stomach in one piece, and then not having it rot from the inside out. So one of the things those kinds of venoms do is digest prey as well as subdue it. Whereas the neurotoxins, in terms of feeding biology, seem to be all about subduing prey, but not digesting it.
Thank you. All right. Well, please join me in thanking Harold, Jeffrey, and Harry.